Hello everyone! Today we are going to learn about the structure of plant stems. The stems and roots of plants are the same, and different plant types have different structures. We will examine the structure of plant stems in three parts: the primary structure of dicotyledonous stems, their secondary structure, and the structure of monocotyledonous stems. First, let's look at the primary structure of the dicotyledonous stem. The primary structure of the stem is the structure formed by the primary growth of cells derived from the apical meristem of the stem. It can be divided into three parts: epidermis, cortex, and vascular column. Let's start with the epidermis. The epidermis is the outermost layer of living cells of the young stem, generally rectangular in shape, with a keratinized outer wall and containing stomata, which is a channel for gas exchange. Next comes the cortex. The cortex is located in the inner part of the epidermis, consisting of multiple cell layers, with chloroplasts often present in the outer layers, so the young stems are often green. It contains parenchyma or sclerenchyma, which enhances the supporting effect. There is generally no endodermis. The innermost part of the stem is the vascular column, also known as the middle column, that is, all the tissues within the cortex, including vascular bundles, pith, and medullary rays. Let's start with the vascular bundle. Vascular bundles are distributed in the parenchyma of the stem, arranged in a circle. Most vascular bundles are collateral, with a few being bicollateral, such as in pumpkin. Each bundle consists of primary xylem, primary phloem, and fascicular cambium in between. Both pith and medullary rays are composed of parenchyma tissue. The pith is located at the center of the young stem, and its cells are larger in volume. The medullary rays are located between the vascular bundles and have the function of transverse transport. This is the primary structure of the plant stem. Most dicotyledonous plants have secondary meristem on the basis of primary growth, namely vascular cambium and cork cambium, which carry out a series of division activities and finally make the stem thickened, which is the secondary growth of the stem. Let's look specifically at how these processes thicken the stem. Let's first look at the production and activity of the vascular cambium. Between the primary xylem and the primary phloem, a layer of cells with the ability to divide is retained and develops into the fascicular cambium, which constitutes the main part of the cambium. A layer of cells in the medullary rays adjacent to the fascicular cambium that restores the ability to divide is called the interfascicular cambium, as it lies between the vascular bundles. The fascicular and interfascicular cambium connect to form a complete cambium ring. The vascular cambium undergoes periclinal divisions, producing secondary xylem toward the inside and secondary phloem toward the outside, adding to the inner side of the primary phloem and the outer side of the primary xylem. At the same time, vertical division increases the number of autologous cells, thereby enlarging the circumference of the cambium ring. Some people compare the process to blowing up a balloon. Now, let's look at the activity of the cork cambium. The cork cambium can arise from the epidermis, cortical parenchyma, collenchyma, and even parenchyma cells in primary phloem and secondary phloem. The cork cambium splits outwards to form the cork layer, inward to form the phyloderm, with the cork cambium itself in the middle. Together, these three layers form the periderm, a secondary protective tissue. Next, we summarize the secondary structure of dicotyledonous stems. The secondary structure of the stem results from the activity of the vascular cambium and cork cambium. From the outside inward, these are periderm, cortex, primary phloem, secondary phloem, vascular cambium, secondary xylem, primary xylem, and pith. There are also radiating rays, mainly medullary rays and vascular rays. Finally, let's look at the structure of the monocotyledon stem and see how it differs from dicotyledon. The stem of most monocotyledonous plants has only primary structure, so the structure is relatively simple. A few have secondary structures, but differ from dicotyledonous stems. Using the stem of grasses, Poaceae, as an example, to illustrate the characteristics of primary structure of monocotyledon stems. The structure of the gramineous plant stem consists of epidermis, basic tissue, and vascular bundle. Epidermal cells are of two types, long and short, with keratinized and silicified walls, etc. The degree of silication has a certain relationship with the ability to resist pests. A small number of stomata are also present on the stem epidermis. The inner part of the epidermis is the basic tissue. In the young stem, there are a few chloroplasts in the parenchyma cells near the epidermis, which can carry out photosynthesis. Compared with the primary structure of dicotyledonous stems, the vascular bundle of gramineous stems is more numerous and scattered in the basic tissues, and there is no clear distinction between cortex and stele. The vascular bundle of most monocotyledonous plants consists of xylem and phloem, without cambium or secondary structure. Vascular bundles are generally arranged in two ways: scattered irregularly throughout the basic tissue, And the cortex and pulp are difficult to distinguish, such as the vascular bundle of corn, sorghum, sugarcane, etc., as shown on the left. Arranged more regularly, often in two rings with the central pith. In some plants, when the stem grows up, the pith may break down to form a pith cavity, such as rice, wheat, etc., as shown on the right. Although vascular bundles are arranged in different ways, the structure of the vascular bundles is similar. All are collateral and closed, lacking a cambium. That's all for today's lesson, and I'll see you next time.